Thank you everybody for coming to my honors thesis defense. Today I will be discussing a lot of the projects that I worked on this past school year, which have to do with plant aphid and parasitoid interactions and how the effects of host plant toxicity and transgenerational stress will affect this system. Before I get into the details about my projects, I wanted to give an outline of what I will be covering today. So as an overview, I will be discussing ecological systems and insect interactions. And then I will move into chapter 1, which discusses the effects of host plant toxicity on aphid host suitability for parasitoid wasps. I will then move into chapter 2, where I will discuss the effects of transgenerational stress on aphid gene expression. And then I will also discuss some broad conclusions over all of my projects. And then at the end, there will be time for some questions. So just as a general introduction to my project, I wanted to discuss some insects and how they interact with their environments. So insects are incredibly diverse, and they are the most diverse class of animal on the planet today. Plants and insects have evolved very close relationships over evolutionary time because of this diversity and the amount of time that they have been evolving. And of course, these relationships between plants and insects are not static. Outside influence of other organisms like predators can affect the relationships between a host plant and a herbivorous insect. And just as an example, I have a picture of a monarch caterpillar feeding on a leaf. And so this is actually an example of a specified interaction, since monarch caterpillars will only feed on certain types of plants. One of the main insects that I will be focusing on in my projects are aphids. Aphids are very small herbivorous phloem feeding insects, so basically they feed on plant saps. Aphids have an interesting reproductive cycle. So for the majority of the year, aphids will reproduce clonally, and they are entirely female. And so a mom aphid, as you can see in this picture, is giving birth live to her genetically identical offspring. And so since they are clonal and they reproduce without any egg stages, the populations can actually grow very quickly in a very short amount of time. In some species, a change in daylight and day length can actually trigger a sexual life cycle, where a generation of males and females will reproduce, lay eggs, and then when those eggs hatch in the spring, the reproductive cycle that is asexual will start over again. Aphids are insects that are commonly predated on by lady beetles, lacewing larvae, and parasitoid wasps. And they are a widespread agricultural pest that can cause hundreds of millions of dollars in damage to crops in just the United States. Another focus of interest for my projects is a parasitoid wasp. And parasitoid wasps are interesting because adults will lay their eggs inside of aphids, where they will then grow into adult wasps. And so the adult wasps are not parasites. The larvae of the wasp are the parasite. And so the larvae will feed on the aphids as they grow and will eventually emerge as adults. During wasp pupation, the aphid becomes what we call a mummy. Here in this picture, you can see that I've circled an example of a mummy. It is basically an enlarged off-color aphid that has turned into a protective husk that keeps the pupated wasp from damage while it develops. Parasitoid wasps are actually one of the most diverse insect groups and are often used as agricultural pest control for insects such as aphids. To give more information on the parasitoid wasp life cycle, I wanted to go over this figure. So here in this top, you can see, as in the picture in the last slide, a parasitoid wasp injecting its eggs into an aphid. And then as time goes on, the wasp larvae will continue to grow and eat the inside of the aphid, which will eventually kill the aphid. After about 10 days, a mummy will form, which the aphid has died at this point and is now just serving to be a protective cover for the wasp. And then between 3 and 5 days later, the wasp will emerge from the casing, and it will eventually be a fully formed adult wasp that will go lay its eggs in new aphids. For my first set of experiments, I focused on milkweed aphids as a toxic host for parasitoid wasps. Milkweed aphids will feed on toxic milkweed plants and can sequester the toxins found in milkweeds that are called cardenolides as a defense against predation. Cardenolides are a steroid chemical that induce damage in cardiac tissue by blocking sodium-potassium ATPases, which would basically induce heart attacks. Cardenolides are toxic for many parasitoid wasps that will attack milkweed aphids, and despite this, they still manage to survive on milkweed aphids as hosts. Additionally, I wanted to investigate how water stress and its impact on milkweed aphids as hosts for parasitoid wasps based on how I water their host plants. So it is known that foliar cardenolide concentrations will increase in the milkweed species A. sierrica under water stress treatment. The milkweed aphids feeding on water stress milkweed plants may therefore have more cardenolides sequestered in their tissues as a result of this. And the increase in cardenolide sequestration could result in reduced suitability of milkweed aphids as a parasitoid host due to the increased toxin level. So based on this background information, I have developed two hypotheses for my future experiments, is that one, milkweed aphids feeding on a more toxic milkweed species will sequester more cardenolides than milkweed aphids feeding on less toxic milkweeds, and milkweed aphids feeding on milkweed plants experiencing water stress will be less suitable hosts for parasitoid wasps than milkweed aphids feeding on well-watered milkweed plants. I want to discuss my study system more in detail before I talk about my direct results. So the plants that I used for my experiments are two different species of milkweed from the same family, Asclepias tuberosa and Asclepias viridis. These plants are perennial flowering plants. They grow across the United States, including most southern states, and are very common because they are planted for monarch butterflies. The first species, A. tuberosa, is characterized by having very little or no cardenolide content in their tissues. A. viridis, on the other hand, has very high cardenolide concentrations in their tissues, meaning they might be a more toxic host, potentially. Milkweed aphids will readily feed on both species of these milkweed plants. The insects and the insects that I will be using are, of course, the milkweed aphids and a parasitoid wasp species, Aphidius colemanni. Milkweed aphids, as I said before, will readily feed on milkweed plants of all species and types, and Aphidius colemanni are known to both attack and survive on milkweed aphids. So the first thing I did to investigate potential relationship between host and aphid, I did a field survey where I collected milkweed plants, milkweed aphids, and parasitoid wasps from Austin College properties such as the Idea Center Gardens and the WCC Gardens, as well as the Edith and Clinton Sneed Prairie and Restoration Site, and all collections were made in September of 2019. All of the plant and aphid samples were frozen as soon as possible at -80°C so that future analysis could be done on them. And mummies were carefully collected and stored in the lab so that we could observe potential future emergences and identify wasp species. The results of our field collections revealed that very few colonies of milkweed aphids were around in this area. In addition, even fewer mummies were found, which basically indicates that very little evidence of parasitoid interactions existed in this survey. The plants that we surveyed in this survey were in panel A, Acorus savica, which is also known as tropical milkweed. It is characterized as a mid to high content cardenolides. And we did find evidence of both aphids and mummies on this species. In the middle, we have A. viridis. And like I said before, it is mid to high content cardenolides. And we found very little evidence of aphids and no evidence of parasitoids on the many species we encountered during the survey. And then in the last panel, we have C. lave, which is actually a viney species of milkweed colloquially known as honey vine. And we also found evidence of milkweed aphids on this species of plant, and very little evidence of parasitoid wasps overall. In the first experiment that I ran in the lab, I was looking at the effects of plant cardenolide concentration on aphid cardenolide concentration. And so in order to do this, I introduced 6 A. viridis plants and 6 A. tuberosa plants with 1 adult milkweed aphid and tracked the population sizes over 10 days. In addition to these plants, 6 A. viridis and 5 A. tuberosa were not assigned to have aphids and were used as a control group. And then after 10 days, aphid and plant leaf samples were collected and frozen at -80°C for future analysis. The results that we found from this can be seen in this figure. So I have average number of aphids on Y and then time and days on the X. And you can see that the number of aphids in the aphid colonies began to grow steadily over time. However, the colony sizes were very similar between both species of plant. For my second experiment, I focused on the effects of plant cardenolide concentration and water stress on aphid cardenolide concentration. So this experiment was set up very similarly to experiment 1. However, I had to use a 2x2x2 factorial design where plants were either assigned water stress or well-watered treatments and either hosted aphids or did not host aphids. And so here in this table, you can see based on each species of plant and the aphid presence or not, how many in each treatment group we ended up having. And in total, we ended up with 8 different treatment groups. What we ended up finding is present here in numbered graph, or first graph for A. viridis. Average number of aphids is again on the Y and time in days is on the X. And similarly to experiment 1, colony sizes grew steadily over time, but did not differ between either water-stressed or well-watered plants. Similarly, in this figure, we can see again that the colonies did grow over time, but they were not different between well-watered and water-stressed A. tuberosa. For my third experiment, I focused on parasitoid wasp host suitability in response to milkweed aphids feeding on water-stressed or well-watered plants. And so tuberosa plants from experiment 2 were actually used in this experiment instead of being sampled for at the end. So I took 5 water-stressed Asclepias tuberosa and 5 well-watered Asclepias tuberosa and exposed these plants with the aphid colonies to 3 Aphidius colemanni wasps for 23 hours. After about 6 days, we begin to see mummies forming on each plant. And so at that point, we counted in all of the mummies on each plant and moved them into individual petri dishes. By moving them into individual petri dishes, we were able to accurately observe how many wasps successfully emerged from the original set of mummies per plant. And so we observed emergences for 3 days after the first emergences were made. And then all the emerged wasps were collected and flash frozen for later analysis. What we found in this first graph, we see average number of mummies per plant on the Y and the watering condition on the X. And you can see that there are no differences between well-watered and water-stressed number of mummies per plant. In addition, very few mummies were formed on both watering treatment plants. Additionally, in the second figure, you can see the percent wasp emergence on the Y and then the watering condition again on the X. And although 100% of the mummies that were on well-watered plants emerged successfully, and only 2 out of the 8 emerged successfully from water-stressed plants, there were no significant differences found between these 2 groups. The last steps in these projects were to take samples from my previous experiments and analyze them for cardenolide concentrations using HPLC. HPLC stands for high performance liquid chromatography, and it basically separates, identifies, and quantifies each component present in a sample. And so in order to prep my samples for this process, I first needed to freeze dry all of my samples to remove excess moisture. And then I would take 0.5 grams of crushed freeze-dried sample, or the entire weight of the sample if it weighed less than 0.5 grams, and mixed it with 1 milliliter of 100% methanol. Methanol worked to extract soluble parts of the sample so that the cellulose of the plant and aphid cells could be left behind. This methanol extraction was done 2 times, and the resulting vials had 980 microliters of extracted sample and 20 microliters of a digitoxin spike. The purpose of a spike is to allow the HPLC machine to get an accurate read of low concentration solutes. And so since we had samples that were very small, we were concerned that the cardenolide concentration wouldn't be recognizable in an accurate way by the machine. And so by adding 20 microliters of digitoxin, which is a chemical that is structured similar to a cardenolide, it just basically gives the HPLC machine something to look for and makes the measurements more accurate. Before the analysis, all of our samples were filtered through a syringe filter to avoid any particulate matter that might damage the machine. And then aphid and plant samples were run as triplicates on the HPLC machine. In order to analyze this data, a 3-way ANOVA was run on samples from only experiment 2 due to time constraints on sample preparation. So the results for this sample 2 cardenolide concentrations are as follows. This graph is very busy, and it is showing the average cardenolide concentration on the Y and then the conditions on the X. And so what we're looking for here is any differences in cardenolide concentrations based on aphid presence, plant species, and watering condition. So on here you can see Asclepias viridis plants are first, and then Asclepias tuberosa plants are second. The little A+ and A- just indicates whether aphids were present or absent. And then in this key, colors are related to whether a plant was well watered or water stressed. We did not find any effects on the aphid present plant species and watering condition interaction for this set of data. We did look at just the watering treatment as an effect, and so on this figure I'm showing average cardenolide concentration on the Y, as well as whether plants were well watered or water stressed on the X. And we did see that plant cardenolide concentrations were higher in plants on average that were water stressed than in plants that were well watered. We also found an effect for cardenolide concentrations and the interaction between aphid presence and plant species. And so on the Y-axis, again, average cardenolide concentration. On the X-axis, I present the different species and whether or not aphids were present or absent in the samples. And so when we did a post-hoc analysis on this set of data, we actually did not find any specific interactions among any of these treatments. And so we are still currently wondering why a significant interaction popped up, but no post-hoc differences can be located. Since the p-value is very close to 0.05, we're wondering if this has some effect on why this data set is reporting differences that we can't find. I also have data on just cardenolide content in aphids. So in this figure, I have the cardenolide concentration on the Y, and then the plant condition again on the X. So A. viridis is once again first, followed by A. tuberosa, and then well-watered and water-stressed, and then again well-watered and water-stressed. And so the aphids appear to sequester cardenolides based on the milkweed species and watering treatment. However, since there was only one sample of aphids prepped per treatment group, no official conclusions can be drawn for this data set. Finally, we wanted to look at how aphid cardenolide concentrations may have correlated with average plant cardenolide concentrations. And so we plotted aphid cardenolide concentrations on the Y and the average plant cardenolide concentrations on the X. We found that plant cardenolide concentrations appear to be relatively correlated with aphid cardenolide concentrations. However, again, since the aphid samples, there was only one per treatment group, no significant correlation can be found here. So now we will be moving on to the Chapter 1 discussion items. And so for my survey, one of the main things that would be beneficial would be to make it longer. A couple of the papers that inspired my survey methods surveyed for a couple years at a time in various places. And although surveying for that long for these purposes would not be logical, I feel like extending the survey from a few weeks to maybe a few months would be beneficial and increase potential encounters with aphids and with wasps. In addition to that, surveying around more local areas like open fields, parks, and neighborhoods may also increase potential interactions. Milkweed aphids are known to be active in the spring and summer, and so instead of surveying in the late summer and early fall, spending more time surveying in June, July, and August may increase encounters with wasps and milkweed aphids. However, previous Austin College student surveys of aphids indicate that milkweed aphids might just be uncommon in local Sherman areas, and so they didn't find very many colonies when they surveyed during the summer months. Moving on to experiment 1 and 2, the aphid populations did not differ between milkweed species of differing cardenolide content, and this basically confirms that this species of aphid is well adapted at feeding on milkweeds with varying cardenolide concentration, which is known already. However, our evidence of similar milkweed aphid colony size across water treatments contradicted the suggestions presented by Huberty and Deno, who suggested that aphid colonies are smaller on plants that are experiencing water stress. And so the main reason for this is that my plants may not have been adequately water stressed, since both A. tuberosa and A. viridis are known to be very drought and heat tolerant plants. Instead of applying a continuous water stress regime, we may have applied a pulsed water stress regime, and this may not seem like a significant difference. However, the types of watering that you induce make a big deal for the insects feeding on the plants, including aphids. So I wanted to talk about how aphids feed. So they feed by piercing a small mouth part into the plant and searching for a phloem tube, which is represented here. Here's the aphid mouth part, and here's the phloem tube. And so essentially, they will just insert this tube into the phloem, and then the phloem will basically be pushed into their bodies. So the feeding relies on the turgor pressure of the plant, which is just essentially how much water pressure is inside plant cells at any given point. And their feeding is actually a passive process. There is no musculature required, and so it is essentially all related to the internal pressure of the plant. And so the turgor pressure of a plant is affected by the watering treatment that is given. So if you apply no water stress to a plant, the turgor pressure will remain high because there's no lack of water in that plant. However, if you introduce continuous water stress to this plant, the turgor pressure will continue to decrease in a linear fashion over time, as water will just continue to become more and more scarce inside the plant cells. As the turgor pressure continues to decrease, an aphid's ability to feed will become hindered to a point where at this point it will not be able to feed on this plant anymore. And so continuous water stress may actually result in smaller colony counts because these aphids are not able to physically feed on the plants. We see a different pattern when pulsed water stress is introduced to these plants because the turgor pressure is moving from very high to very low depending on the periods of water addition and drought. And so what you end up seeing is a lot of time where the turgor pressure is changing. However, it might still be accessible for an aphid to feed. So why does pulse water stress benefit insects? So by adding a water stress event, cells will start accumulating solutes, and eventually the plant becomes more nutritious for herbivorous insects because nutrients like nitrogen become more concentrated in the plant cells. And so the main takeaways for this are that pulsed water stress results in periods of increased nutrient concentration in a plant, and then periods of turgor high enough for herbivorous insect to feed. And so plants experiencing pulsed water stress may be more nutritious and still accessible to aphid colonies. Future experiments may measure changes in plant nutrients and how it affects aphid colonies in this way. In experiment 3, we did find very few mummies formed on milkweed aphids, which generally indicates that they make a poor host even though a colony could survive. By looking for the eggs and larvae inside of aphids, in addition to mummies that form, would provide more information about how milkweed aphids are suitable hosts for these wasps, since we could find out at what life stage many of the wasps might be dying before they reach the mummy stage. By having a larger number of replicates, we would be able to potentially investigate differences in number of mummies formed based on water condition, and also the successful emergence of adults in relation to water condition. Similarly, including A. viridis to these experiments would expand the study to include a more toxic milkweed species and investigating how that might affect the whole system. Finally, discussing microdenolide analysis. So plants are known to become more toxic during times of water stress. So a result that water stress did affect plant cardenolide concentrations was expected. However, the found interaction between aphid presence and plant species is harder to explain. Martel and Malcolm actually found that a high cardenolide milkweed species, Asclepias curassavica, responded to aphid colonization in a few different ways, whereas a low cardenolide species, Asclepias incarnata, did not respond at all. Asclepias curassavica responded to aphid colonization by first reducing cardenolide content in the very beginning of the aphid colonization, and then as aphid population grew, it began increasing cardenolide
Hey, Nicole, we might be experiencing an audio problem.
Is on the high cardenolide.
Nicole, can you hear me?
I can. Can you hear me?
Yes, I think it might be better now.
Okay.
Could you go back to explaining, I think it went out about when the changes in the cardenolide concentration in the A. curassavica in relation to the aphid population?
Okay. So we're trying to find potential connections with why we saw a significant interaction between plant species and aphid presence. And so Martel and Malcolm had a high species or high cardenolide milkweed species, A. curassavica, and a low cardenolide milkweed species, A. incarnata. What they found was that A. curassavica, the high cardenolide milkweed species, responded in different ways to aphid colonization, but the low cardenolide species, A. incarnata, did not respond to aphid colonization whatsoever. And what the high cardenolide milkweed species was doing was altering cardenolide content based on aphid colonization. So in the beginning of an aphid colonization, cardenolide content would be reduced. And then as aphid populations began to grow, it would increase cardenolide concentrations in plant tissues exponentially. And so the high cardenolide concentration plants are essentially discouraging these aphids from feeding on these plants. And since we also used a high and low cardenolide milkweed species in our experiment, it would be interesting to understand whether our plants are responding in a similar way to the way that Martel and Malcolm found their plants to be interacting. Additionally, the cardenolide concentrations detected in aphid samples seem to align with plant cardenolide concentrations. Some final thoughts on these experiments before we move on to my next set. Overall, larger sample sizes and more replications for all experiments would decrease a lot of the within treatment variation that I was seeing. In addition, conducting HPLC analysis on wild plant samples would be helpful because it would indicate whether the lab-reared plants of the same species had similar or comparable cardenolide concentrations. Going off of that, running these types of experiments in natural settings would seriously benefit our understanding of how these systems work, since a lab setting may overlook a bunch of different interactions that might be existing in a natural environment that can't be replicated in a lab. In addition, further investigation of the effects of water stress on parasitoid excess is important, since it is not known whether parasitoids have evolved any particular mechanism to combat cardenolides in their tissues, or by sequestering them, or even if they can metabolize them, it is not known. Finally, conducting HPLC analysis on parasitoid wasps would allow us to find out if parasitoid wasps are in fact sequestering cardenolides in a similar way to aphids. I will now be moving on to material for my chapter 2. Chapter 2. So this project's focus on genetic effects of parasitoid wasp stress in pea aphids, which is a different species of aphid. So aphids respond to the threat of predation by falling off of their plants, kicking and cornicle secretions. And a cornicle is a protrusion on the dorsal abdomen of aphids, and it would be located right around here. And many of the cornicle secretions are actually toxic and can ward off predators and let them know that they should not be attacked or eaten. These threats, or these responses to predation threats, are energetically costly, and so there is a trade-off between the level of the threat and the severity of the stress response. Pea aphids, which are pictured in this photo here, are a model organism and are well known for producing winged offspring in response to a stressful event like predatory threat. And so in this picture, you can see this is an adult winged aphid and her offspring. And so whether or not these aphids grow up to have wings or they don't, they will be still genetically identical to this mother. So something in gene expression is causing this change. So the paper that inspired this project focused on changes in cuticular protein gene expression in pea aphids, in the adults and the embryos, in response to crowding stress and stress induced by lady beetles. And so they did focus on a predator that was actually physically eating these aphids. And so in this figure, you can see the representation of the expression of cuticular protein genes, the upregulation and downregulation in antennae, head, the gut, cuticle, embryo, and legs. And this is just showing how upregulated, how downregulated, or how relatively similar these genes remained in comparison to a control gene. And so you can see that most cuticular protein genes were upregulated in the leg, upregulated in the embryo, and were either downregulated or remained similar in the antennae. But CP23 was downregulated in embryo and upregulated in the antennae, which was interesting to me when I was looking through this paper, deciding on what to investigate. And it is currently unknown if the stress response that is seen in this project occurs also in response to parasitoid wasp stress, and if it persists beyond the directly exposed generation. So no investigation has been done on aphids offspring that have been exposed. So my two hypotheses for chapter 2 are: one, that cuticular protein gene expression will differ among pea aphids exposed to parasitoid wasp stress, and two, this gene expression change will persist for longer than one generation after the exposure. So in order to run these experiments, the plants that I used were fava beans, and these are vine-like plants that grow very quickly and are commonly used in experiments like this to host insects. They are not like milkweed plants and do not produce any toxins, and they do readily host aphids like pea aphids. I will also be using, of course, pea aphids and then a parasitoid wasp species, Aphidius airbi. Pea aphids will, as I said, readily feed on bean plants like fava beans and are known to be an attractive host to the wasp Aphidius airvi. Aphidius airvi are similar to Aphidius colemanni, the wasp used in my previous projects. However, they have a slightly larger body size. So this wasp does have the exact same life cycle and reproductive ability as was previously shown. So in order to set up this experiment, I needed to stress aphids while also keeping them alive so that multiple generations could be tracked. And since Aphidius airvi will kill these aphids, I needed to figure out a way to expose them while also keeping them protected. And so in one enclosure, I had a fava bean plant with 30 adult pea aphids on it. And it was covered with an enclosure that had mesh openings on the side. Directly next to this plant, I placed a second bean plant with 30 adult pea aphids. However, this plant would be sacrificial since the wasps would attack these aphids. The plan was that any volatile cues of stress and potential cues that the wasps would release would transfer between the mesh openings in the covers so that these aphids would be adequately stressed without being attacked by the wasps and dying. In addition to these two plants, I set up a third plant that was also next to the wasp plant, where I wanted to track the dropping behavior, which I would use as an indirect measure for the amount of stress that the aphids were experiencing. And so the plant was set up in the exact same way as the two previous plants. At the bottom, there would be a sticky card where if an aphid were to fall, it would get stuck and would not be able to crawl back up the plant. And so at the end of the exposure, when aphids were being collected, I could see how many dropped in response to the stress being induced over here in this plant. And so for this experiment, we set up 4 of these replicates for each treatment group. So aphids either exposed or not exposed to these wasps. And then 2 more generations were followed with this exact same setup, except that the aphids in generations F1 and F2 were not directly exposed to wasps in the way that the first generation was. And so the initial results that we found from this experiment have to do with offspring production and dropping. And so in this first figure, I'm showing the average number of offspring and the generation, so F0, F1, and F2. We did find that the offspring production was lowest in generation F2, regardless of the stress treatment. Additionally, I have a graph with average number of drops on the Y and then generation F0, F1, and F2 on the X. And dropping behavior was similar across all the generations, regardless of the treatment group once again. In addition to collecting information on dropping and offspring, we needed to prep samples of aphids for qPCR. The purpose of doing qPCR is that I would be able to quantify the expression of a target gene in my isolated tissues. And so RNA was extracted from both aphid legs and antennae. In this table, I am presenting the primers that we used in this experiment for our target genes. And these were also taken from the WHO paper that was discussed earlier. So we chose to look at CP17, CP38, CP23, which was the first gene present in the heat map that I discussed earlier, and then at the bottom is our reference gene. Unfortunately, I was not able to finish this experiment. We did validate primers and aphid samples were collected and I was able to dissect them, but only the samples from generation F0 had any RNA extracted. Due to the current pandemic restrictions on lab work at the college, I was not able to extract RNA from F1 or F2, and the qPCR was not due to time constraints and restriction of lab work. Now I will be discussing the chapter 2 results that I did find. So we did find a smaller number of offspring in F2 as compared to the other generations. And this is very likely due to the fact that fava beans grow very quickly. And depending on how old they are before they are used in experiments, they can outgrow the enclosures that we use very quickly. I observed in summer research that when the fava beans begin to outgrow the enclosures, the humidity inside of the enclosures rises dramatically, which ended up killing a bunch of the aphids. And so populations may have been affected by excess humidity in plants that were not correctly sized for the enclosures. Additionally, we didn't find very many drops across generations. It was low and not different. And this is likely because the aphids in my experiment were not adequately stressed in generation F0. And so we relied on the olfactory cues to induce the stress that we wanted to see. However, it might be true that olfactory, visual, and tactile cues might be important for P. aphids to recognize a potential predatory threat. Cooter et al. Was investigating aphid stress responses while being exposed to lady beetles and aphid lions. This is a picture of an aphid lion, and it is basically another way to refer to a lacewing larvae as, yes. So the experiment, they sprayed aphid lion remains on the base of a plant, and this was a significant enough stress to induce green peach aphids to begin dropping off their plants. And so it might just be true that the olfactory cues given off by parasitoid wasps are very different from that of like lady beetles and aphid lions. And so investigating more ways of exposing these aphids to other types of cues would better induce the stress. One way that I believe would increase the success of this experiment would be to use very large aphid colony sizes so that ARV could be directly introduced to the colony and forage freely. And so there would still be enough aphids at the end of the exposure to use in future experiments. Since this project was unfortunately left unfinished, there are a couple of future directions that I feel are very important still. The effects of the parasitoid wasps on changes in expression of cuticular protein genes in P. aphids are still not known and still need to be investigated. Additionally, studies investigating changes in cuticular protein gene expression have only currently been conducted in one generation. To wrap up all of my work with a few conclusions, the interactions between parasitoids, host aphids, and host plants are very complex. There are a lot of different types of experiments that need to be done to understand the different parts of what is going on in relation to these, specifically this system. Additionally, watering conditions do change the plant physiology, which can impact insect communities, including predators such as parasitoid wasps. A focus on the direct effects of cardenolides on parasitoid wasps is still necessary since, again, there is no information about how parasitoid wasps deal with the toxic cardenolides in their host aphids. Finally, evidence for genetic changes in more than one aphid generation would enforce the understanding that aphid gene expression is plastic. Even though aphids may have wings, that doesn't mean that they are genetically different from an aphid that doesn't have wings. And so understanding how cuticular proteins fall into that category is important. At the end, I would like to acknowledge Austin College's summer research program, the Austin College Biology and Chemistry Department, and the Departmental Honors Program Committee for giving me all of the equipment that I needed and funding my projects and giving me great spaces to do everything that I need to do. A special thank you to my project mentor, Dr. Garcia, for working very closely with me in the summer and the school year, making this a very fun, very informative experience for me. I feel like I've gained so much from working with her very closely. My lab partners, Matt Lee, Sarah Joseph, Anusha Jacob, and Kenny Ota, spent a lot of time this semester and last helping me out with my projects and making sure I was able to get everything done. My thesis committee, Dr. Aiello and Dr. Hebda, I thank you for working with me this year on this project. And for Dr. Aiello, I really appreciate all the help I got on my molecular projects. I'd also like to thank Dr. Glenn, who helped me out a lot with learning how to do HPLC and figuring out what I needed to do with the machinery and allowing me to come up into the chemistry department and use what I needed. And thank you to Dr. Richardson for working with me on learning how to use the freeze dryer and giving me access to his equipment in his lab. And now I have time for some questions.